This is Chapter 5, the cell cycle. The cell cycle is a process of cell division that serves to increase the number of somatic cells. So you're asking me, what are somatic cells? Somatic cells are body cells, which exclude cells that undergo meiosis and become sperm or egg cells. I think it's very interesting that every day your body produces millions of new red blood cells, skin cells, or cells that line your respiratory and digestive tract. In addition to growth, cell division is involved in the repair of tissues after an injury. So, the cell cycle. While cell division increases the number of cells in the body, the process of apoptosis or programmed cell death acts to decrease the number of cells. And that's important. There's actually, I think it's very interesting that you were born with webbing, or not born, I'm sorry, you were developed inside of your mother's womb with webbing in between your fingers and your toes. And through apoptosis, we actually have death of these cells, which allows our hands to look like they do today. Another example of this is the tail of a tadpole as it disappears as it matures into the frog. Cancer, some types of cancer develops due to a failure of apoptosis. And if you think this kind of makes sense, whenever your cells in your body begin to be mutated or there's something that goes wrong, your body has a built-in mechanism that tells your cells, hey, something's going wrong. There's checkpoints within the cell cycle during the cellular division that triggers apoptosis and triggers programmed cell death. If you have a failure of programmed cell death, this is going to predispose your cells to continue to divide. Not only are rapidly uncontrolled dividing cells a bad idea, but they're going to be full of mutations. And the fact that they're full of mutations are going to lead to cancer. So here, cell division is part of a cell cycle. As you can see here, we have the entire cell cycle. This is called interphase, and interphase is going to be very important. Through interphase, we are going to have 3 different points. We're going to have G1, which is growth phase 1, in which cells are going to rapidly double their organelles, and then they're going to go on to the S phase. That is, if they pass this checkpoint. There's a checkpoint here that checks to see if all of your cells have the right amount of DNA. And if they do not, then they'll undergo something called programmed cell death or apoptosis. If they do, hey, everything's good. Let's move on to the S phase. Within the S phase, this is where DNA synthesis occurs. DNA synthesis will then move on into the G2 phase, and this is where rapid cell growth occurs again. So your body is going to spend 90% of its time within interphase. However, if there is no checkpoints here, we are going to have rapid division of cells, or we're going to allow cells to get through that has mutations. Here we have the mitotic phase, and in the mitotic phase we have mitosis, which is the formation of 2 daughter cells, and that's important to know. And then we have cytokinesis, and cytokinesis is the division of cytoplasm, which is used in order to produce 2 new cells. So as you can see here, the cell cycle is an orderly set of stages that takes place between the time a cell divides and the time resulting cells also divide. So again, we said, how does this cell cycle work? So most of the cell cycle, like we previously just said, is spent in interphase. And interphase is just the phases that make up the G1, the S, and the G2 phase. And again, 90% of your body is going to be spent in interphase. So here we say here that cells normally go through a cycle that consists of 4 different stages: G1, S, G2, and M. And the M stage stands for mitosis. There is a G0 stage, this is just going to represent a holding stage for cells that are not undergoing cell division. There's actually cells within your body that are constantly in G0, and they're meant to stay in G0 and not undergo division because essentially they're not needed. What are examples of this? Nerve cells. There's really no good reason for your nerve cells to continue to grow. However, hair is rapidly dividing, at least for most of us. But skin is rapidly growing if you're ever burned, and for example, a sunburn, and your skin begins to peel as you peel off that new layer. It would be bad if our cells were stuck in G0 and unable to produce that new layer of skin. But we know that's simply not true. It does. But nerves are not really needed once they are formed. Muscle, for the most part, muscle spends most of its time in G0 unless you are stimulating the muscle through growth factors like growth hormone and things like that. Then it will undergo interphase of mitosis. So we say here, how does the cell cycle work? So the cell goes through a cycle that includes interphase, again, these 4 phases that we previously discussed, and cell division. And we know that cell division consists of mitosis and cytokinesis. And we know that cytokinesis is just splitting of the cytoplasm. And we'll discuss these phases in a little more detail. And again, these are important to know what occurs in each of these phases as they will be tested. G1 phase, there's growth as the cell prepares to divide. Again, we say there's doubling of the organelles in order to have enough material to undergo DNA synthesis. So we know that DNA synthesis occurs within the S phase. This is where DNA replication occurs and where we have the formation of something called sister chromatids. And we'll kind of go over that in a little more detail. The M phase, when daughter cells receive a full component of chromosomes. So we know that within the S phase, this is where DNA synthesis occurs. We know that this is where the sister chromatids are formed. And within the M phase, we have a full complement of chromosomes. So here we'll talk about the G1 phase a little bit more. This is where cells can either, so we'll just start out. During the G1 phase, we say that cells double its organelles. And we know that cells need to double the amount of organelles they have in order to have enough genetic material to undergo DNA synthesis. And that actually makes a little bit sense. So if for some reason that we don't progress onto the S phase, we don't have enough number of organelles, we can trigger something called apoptosis, or we can undergo something called quiescence. And quiescence is just G0 when everything is quiet down. And when things are quiet down, they are not undergoing cell division. Differentiation. Cellular differentiation is just the changing of cells from one specific type of cell to a different. This could be the differentiation of cells from a hair cell to a skin cell, or something along the lines of that. So here we say that some cells are said to have entered a G0 stage or a pause during the cell cycle. And examples of that we said previously are nerve and muscle cells. They continue to perform their normal functions, but no longer prepare for cell division. The S phase. So we have the proper amount of genetic material. So let's move on to the S phase. And we know that within the S phase, this is where DNA synthesis occurs. And we know that because DNA synthesis occurs, we can have sister chromatids. A little bit of a tongue twister for you. During the S stage, DNA replication occurs. We've said that. At the beginning of the S stage, each chromosome is composed of one DNA molecule, which is called a chromatid, and this is important for examination purposes. The chromatid, following replication, a chromosome consists of a pair of sister chromatids held together at a centromere. And you guys remember the centromere? What it's composed of? That's right, a microtubule. And it's because of this microtubule, and we know that microtubules are responsible for movement, that we can have separation here in a minute. One of two genetically identical chromosomal units that are as a result of DNA replication and are attached to each other at the centromere. At the end of the S stage, each chromosome consists of two sister chromatids that have identical DNA sequences. Another way of expressing this is to say that DNA replication has resulted in duplicated chromosomes. The sister chromatids remain attached until they are separated during mitosis. And we know that this separation is able to occur because the centromeres consist of microtubules, and the microtubules are responsible for movement. So this is what allows this separation. So here we see that we have the G1 phase, and during the G1 phase, we're going to undergo transcription, and we know that transcription occurs after transcription is going to help with the process of DNA synthesis. And after we have DNA synthesis occurring here, we're going to form the chromatid, which is just a chromosome consisting of a pair of sister chromatids. And we know that sister chromatids are just genetically identical chromosomal units. So after the S phase, we're going to undergo the G2 phase. And during the G2 phase, the cell synthesizes the proteins for cellular division, such as the proteins that make up some cells, such as nerve and muscle cells. While in the G0 stage, the cell continues to perform normal everyday processes, but no preparations are being made for cell division. Cells may not leave the G0 stage without proper signals from other cells and other parts of the body. So all I want you to know is during the G2 stage, the cell synthesizes proteins for cell division. So to sum it up, G1, there's growth as the cells prepare to divide. S phase, S stands for DNA synthesis. And the G2 phase, more growth occurs. During M phase, daughter cells receive a full complement of chromosomes. Cytokinesis is just cellular division. That's all you have to know for my exam. We know that there is a mitotic stage following interphase, following G1, S, G2. We are going to go into the mitotic stage. So this stage includes not only mitosis, and we know that mitosis stands for the division of the nucleus, and the nucleus contains what? That's right, the genetic material. This is actually where interphase is occurring. But also cytokinesis must occur. So during the mitotic stage, we have cellular division, dividing of the cells, but we also have cytokinesis, which is dividing of the cytoplasm. So we say here the cytokinesis is the dividing of the cytoplasm, which may not occur in all cells. So we say here that during mitosis, the sister chromatids of each chromosome separate. We know this separation occurs because they are attached at the centromere. And because they're attached at the centromere is made of microtubules, they're able to remove. But getting back to this, these sister chromatids separate, becoming daughter chromosomes that are distributed to two daughter nuclei. Let's talk about control. These are important points, and I want you to know these as they will be on the exam. There's three important checkpoints. Again, we say here in G1 prior to the S stage. We want to know, is there enough genetic material to move on to the S stage for DNA synthesis to occur? Is there any mutation in this DNA that's been produced? And there's a very, 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 very important, it's the control checkpoint of the cell. This is known as P53, and this will be on your exam. The P53 is responsible for detecting DNA damage. This stops the cell cycle. P53 protein is active at the G1 checkpoint, and if DNA is damaged and cannot be repaired, this protein initiates apoptosis. So again, just to reiterate this, you have here G1. If there's DNA damage, if you've been exposed to radiation, radiation can induce DNA damage, or if you've cigarette smoke can induce DNA damage. The cells that are dividing, let's say they're in the lung, if P53 senses that DNA damage, it can trigger something called apoptosis, and apoptosis will then kill the cell. If for some reason that P53 is mutated, and there's actually a syndrome called Leaf-Ramani syndrome. Leaf-Ramani syndrome, there is a p53 mutation. Because there is a p53 mutation, p53 is unable to detect DNA damage. It is unable to stop the cell if there is DNA damage. So what do you think would occur? That's right, there will be rapidly, uncontrolled dividing cells that may or may not have mutations. These people get cancers of various different kinds. Another checkpoint is at the G2 phase, prior to the M stage and near the end of mitosis. But the one I want you to know is P53. I want you to know about P53. Other important controls of the cell cycle or growth factors, again, we said that nerve and muscle cells are consistently in the G0 phase, they're in the resting phase. However, during times of increased activity, during sleep, we know that sleep is responsible for the initiation of growth hormone. That's why they say it's important to get a lot of rest when you're younger, when you're growing. That's because during your sleep, growth hormone is secreted from the hypothalamic pituitary axis, from the HPA axis, and growth hormone is going to act as a growth factor within the cell cycle, and that's going to allow your muscles to undergo interface. So you've got muscle cells that are in G0, growth hormone is secreted, it's going to move the cells from G0 back into G1 and initiate control of that cell. So growth factors are example of external signals that control the progress of the cell through the cell cycle. Finishing up, there are genes. Genes control the cell cycle, and I want you to know both of these terms for examination purposes: proto-oncogenes and tumor suppressor genes. Tumor suppressor kind of sounds like its names. These genes inhibit the cell cycle. So tumor suppressor genes, when these genes mutate, their products no longer inhibit the cell cycle. So if you have a mutation in P53 and BRCA1 or BRCA1, this gene is responsible for breast cancer, as you can see here, BRCA1. If you've ever heard of women who have a family history of breast cancer, they may test them for the BRCA1 or the BRCA1 tumor suppressor gene. And if this gene is mutated, it makes sense that you can't inhibit the cell cycle. Therefore, you're going to have rapid growth of breast cells. We know that cells make up tissue, and tissue, whenever you have rapid growth of tissue, you have something called tumor formation. And then you have proto-oncogenes. And in order to understand oncogenes, you have to understand proto-oncogenes. So proto-oncogenes are genes that encode proteins that promote the cell cycle and prevent apoptosis. So we know that tumor suppressor genes are going to initiate apoptosis, right? We said that P53's major function is to check for DNA damage. If there is DNA damage, it initiates apoptosis, so that is preventing a tumor by initiating apoptosis. So proto-oncogenes are genes that actually want the cell cycle to go through. So RAS is an example of a proto-oncogene. RAS is going to be responsible for allowing this cell cycle to continue. If you have a mutation in RAS, then it becomes an oncogene. You take away the proto. I think of proto as promoting the cell cycle, but if there's a mutation in the proto-oncogene, it then just becomes an oncogene. We know that oncogenes are going to promote growth of the cell cycle, and it's going to continuously tell the cell cycle to keep going in a circle. These are terms that I want you to know: diploid and haploid. Diploid means there's two sets of chromosomes. So we know that there are 23 pair of chromosomes, right? 23 pair. 23 times 2 is what? 46. So we have 46 chromosomes. We are known as diploid. If you wanted to say what is during meiosis, you have the formation of what's known as haploid number of chromosomes, because you're either producing a sperm or an egg during meiosis. So haploid means half. You get 23 pair of your chromosomes from your mom, 23 from your dad. So Haploid is the 23, and whenever you get the other copy from the other parent, it then becomes diploid. And we'll see that here. Each species has a characteristic number of chromosomes. The total number of chromosomes is diploid, 2 in number, and half this number is haploid. Among eukaryotes, the cell division involves nuclear division and division of the cytoplasm, cytokinesis. Replication of the chromatids precedes cell division. Again, replication of the chromatids, replication of the DNA precedes cell division. The duplicated chromosome is composed of two sister chromatids held together at the centromere. During mitosis, the centromere divides and the daughter chromosomes go into a new nucleus. And during mitosis, these are four important stages that's going to occur, and I want you to know what happens in each of these phases as they will be tested. So during prophase, the chromosomes are going to condense with chromosomal condensation or shrinking of the genetic material. In prometaphase, we begin to form something called a spindle fiber, as you can see right here. In metaphase, this is the most highly tested, the one I'll probably test you on. This is when the chromosomes are aligned at the metaplate here. They're in the perfect here. So just to reiterate this, you have chromosomal condensation, right? Because we are going to begin to divide, the chromosomes are becoming inactivated because there's no more DNA replication occurring right now. So after this, we are going to have beginning to form the spindle fiber. The spindle fiber is going to be, if you guys can remember from previous chapters, the spindle fiber is going to be composed of what? That's right, microtubules. And we know that microtubules are responsible for what? Movement. And then we have metaphase. And during metaphase, we have formation of the metaplate. Whenever you think of anaphase, I want you to think apart. Apart. When the chromatids separate, becoming daughter chromosomes that move toward the poles. So you're going to have separation of the chromosomes during anaphase. That may be an examination question. Two good possible questions for your exam could be, when are the chromosomes aligned at a plate? That would be during metaphase. Or I could ask something like, when the chromatids separate and move to different poles, which phase is this? That would be anaphase. So when new nuclear envelopes form around the daughter chromosomes and cytokinesis is well underway, that occurs during telophase. And during cytokinesis, we know that this is just splitting of the cytoplasm. This occurs differently in animals and plant cells. I just want you to be familiar with the name cell plate, which occurs during plant cells, and cleavage furrow, which occurs during animal cells. Here I have a kind of complicated slide that just tells you how and why we become unique in our composition as humans. And this is through things called crossing over and independent assortment. So crossing over is going to occur after you have formation of the chromosomes. And this, I want you to think of everybody bringing their favorite snack to school and everybody exchanging parts of their lunch for other different parts. So let's say you have 10 friends and 10 friends bring 10 different things. If everybody exchanges different portions of their lunchbox, you're going to get a unique meal at the end of the day. And this is kind of what happens here. You have two different parts of this chromosome. You can see here the X's represented here, and you have the Y's represented here, and you have the different you have a capital Z and a lower Z. As you can see here in these pictures that you're going to have just once crossing over occurs during meiosis, you're going to have an exchange of genetic material in which this chromosome may cross over with this one. And you've got this interwoven, unique portion of genetic material that is going to occur. And independent assortment also occurs during meiosis. So meiosis utilizes two nuclear divisions. During meiosis 1, homologous chromosomes, we know that homologous just means same, undergo synapsis. And crossing over between non-sister chromatids occur. When the homologous chromosomes separate during meiosis 1, Each daughter nucleus receives one member from each chromosome. Therefore, the daughter cells are haploid. That means there's half. Interkinesis during meiosis 1 and 2 does not include DNA replication. Then we have distribution of the daughter chromosomes in order to form a total of 4 new cells. We won't go into too much detail about this. Instead, I want to skip down here to the human life cycle. So, the human life cycle involves both mitosis and meiosis. Mitosis ensures that each somatic cell, and remember what we said somatic cells were, these were just cells that were not egg or sperm. These were actually that composed of different tissue, has the diploid number of chromosomes, right? So, mitosis ensures that each somatic cell has diploid set, so 2N, so 46. And humans, meiosis is part of spermatogenesis, or the formation of sperm, and oogenesis, or the formation of an egg. I want you to know both of those terms, spermatogenesis and oogenesis. So, meiosis, I want you to think of spermatogenesis and oogenesis. This is going to be the formation of sperm and an egg. So, spermatogenesis in males produces 4 viable sperm, whereas oogenesis in females produces 1 egg and 1 polar body. And a polar body is just going to be inactivated DNA that is actually not usable. So, oogenesis does not go to completion unless a sperm fertilizes the secondary oocyte. And that secondary oocyte is then going to begin the process of reproduction in which mitosis can undergo cellular differentiation. So, fertilization produces a zygote, which grows by mitosis. So, again, during meiosis, we're going to have a haploid number of chromosomes. This is important to know. You're going to have 23N. In order to have 46N, in order to have 2N, or 2 times 23 being 46 chromosomes, we have to have fertilization. And what is fertilization? Fertilization is in which the process of an egg and a sperm occurs. So meiosis produces spermatogenesis. It produces 4 viable sperm. Oogenesis is going to produce what? 1 egg and 1 polar body. We know that if fertilization occurs, that means a sperm meets an egg. We know that we can then begin to undergo mitosis and form a zygote. Thank you so much.